to our last speaker for this session. That's Astra Bryant. So she's a senior postdoc at the UCLA in California, and she's working at the interface of parasitology and neuroscience, studying the neural basis of parasitism in third transmitter intestinal parasitic worms. So Astra, the stage is yours.
All right. Hi, everyone. Folks can see us? Okay. Perfect. Okay, so as you heard, I'm Astra, and today I'm going to be talking about my postdoctoral research attempting to understand the neural mechanisms that underlie temperature-driven host seeking in the skin-penetrating human threadworm, Strongyloides stercoralis. So these are intestinal parasitic worms that infect approximately 610 million people worldwide. Infections are often asymptomatic, but heavy infections can lead to chronic gastrointestinal distress, stunted growth and cognitive impairment in children, and even death. Current treatments exclusively seek to reduce worm burdens through anthelminthic drugs, but this strategy is often ineffective due to really high reinfection rates. In addition, there's this growing concern about the rise of drug-resistant parasites. So in the Hallam lab, our hope is that a better understanding of how parasites find their hosts could lead to new therapeutic strategies for nematode control. So Strongyloides stercoralis are infective exclusively during a soil-dwelling, developmentally arrested third larval stage that's very similar to the C. elegans dower stage. So we know that these IL-3s, which are 0.5 millimeter long, can respond to multiple host-emitted sensory cues, including heat. But when I started my postdoc, we didn't really understand the role that thermosensory cues played in driving host-seeking behaviors. And we also had basically no understanding at all of the molecular or cellular adaptations that might underlie parasite-specific thermosensory behaviors. So to address the first question, I tested the migration patterns of IL-3s exposed to temperatures ranging from below ambient to above human body temperature. And I found that they engage in two distinct modes of behavior. Positive thermotaxis, a behavioral mode that we can characterize as attraction to mammalian body temperature, and negative thermotaxis, defined by migration towards temperatures below ambient. And so here I'm just showing you the tracks of individual IL-3s engaging in either positive or negative thermotaxis. And our thinking is that worms that engage in positive thermotaxis are engaging in host seeking, whereas worms that perform negative thermotaxis are engaging in environmental navigation. So because it's so close to Halloween, I thought I might show you a creepy video of the parasites engaging in temperature-driven host seeking. So you're gonna see like 1,000 IL-3s crawling on a linear thermal gradient that ranges from 20 degrees below ambient to 34, which is about the temperature of human skin. So each IL-3 is 0.5 millimeter long. It's gonna look like a black dot. So there you go. And you can see these worms are moving really fast. Like I've sped the video up, but they're traveling 6 centimeters in under 3 minutes, which is pretty good for a worm that is 0.5 millimeter long. And this is the most robust sensory-driven behavior we've ever seen in any worm species. Okay, so what are the molecular and cellular mechanisms that might underlie this behavior? To answer this question, I took advantage of our classic nematode model system, C. elegans. So the molecular and cellular basis of C. elegans thermotaxis has been studied pretty extensively. And stercoralis and C. elegans share many of the same genes, and they both have these really highly compact neuroanatomy that's consistent between individual worms. But despite the broad similarities between these two species, their thermosensory responses are very divergent. So C. elegans avoids temperatures above 26 degrees, as opposed to being attracted to human body temperature. So the first question I ask is, are these divergent behaviors dependent on similar molecular pathways, or have the parasites evolved new pathways? So in C. elegans, thermosensation within their physiological temperature range is primarily mediated by a cyclic GMP signaling cascade. And there's other mechanisms, including trip channels, that contribute to the avoidance of noxious temperatures, which again includes, for C. elegans, anything above 25 degrees. So I used CRISPR-Cas9 mutagenesis to disrupt a key component of the cyclic GMP pathway, the Tax4 gene. So Tax4 encodes a cyclic nucleotide gated channel subunit that in C. elegans is necessary for sensory transduction in general and is specifically required for temperature-driven migration. So when I tested Tax4 mutant IL-3s, I found that they showed significantly reduced positive thermotaxis compared to control worms. And again, here I'm showing you the tracks of those worms, as well as a quantification that shows that Tax4 worms don't sort of migrate as far up the gradient. So this tells us that IL-3 heat-seeking reflects an adaptation of molecular pathways for C. elegans thermotaxis navigation, as opposed to noxious temperature avoidance. In both C. elegans and stercoralis, Tax4 is expressed in multiple head sensory neurons. And in C. elegans, the list of Tax4 positive neurons includes several that contribute to thermosensation, including the primary thermosensory neuron pair, AFD. So I wanted to know, in IL-3s, does the attraction to host body temperature depend on the stercoralis AFD homologs? Or have the parasites co-opted other neurons? So to answer this question, I first had to identify the stercoralis AFD neurons, which turned out to be like a 20-year-old mystery in the field of parasitology that I solved using a genetic approach that I don't have time to tell you about today. And then what I'm doing is I'm going to use a chemicogenetic silencing technique to test if AFD activity is required for IL-3 heat seeking. And it is. So you can see here that worms in which AFD has been silenced don't engage in positive thermotaxis as much as the control worms. Okay, so we have this situation where a common sensory neuron, AFD, contributes to both C. elegans and stercoralis thermotaxis, despite dramatic differences in their behavior. So are those behavioral differences at all reflected in how the AFD of each species responds to thermal stimuli? For example, we might hypothesize that stercoralis AFD could be specialized for encoding warmer temperatures than C. elegans AFD. So here I'm showing you average temperature-driven calcium responses in both C. elegans and stercoralis AFD neurons. So for the aficionados, for both species, I'm using the ratiometric calcium indicator chameleon expressed in AFD neurons only. So what we can see is that for both C. elegans and stercoralis, AFD shows increased activity when I deliver a temperature ramp ranging from below ambient to human skin temperature. There are some really interesting differences, right? For example, as other labs have shown, C. elegans AFD primarily encodes a temperature threshold. So it's not active below ambient temperature, and then it is strongly active in a manner that doesn't clearly differentiate between temperatures that are higher than the ambient. In contrast, for stercoralis, we see that neuronal activity continues to increase almost linearly at temperatures above the threshold. And this suggests to us that stercoralis AFD may be tuned for encoding a broader range of temperatures than C. elegans, including human body heat. So how might these differences arise within the primary sensory neuron? In C. elegans, 3 receptor type guanylate cyclase genes are selectively expressed in AFD and contribute to the transduction of thermal cues through that cyclic GMP pathway I mentioned earlier. So we identified 3 Strongyloides genes that by sequence homology appear to be homologs of the C. elegans genes. And we hypothesized that maybe there are functional differences in these AFD-specific RGCs that might explain the differences in the AFD responses between the 2 species. So what do these RGCs do in C. elegans? Work from Pialis and Gupta's lab at Brandeis showed that in C. elegans, 2 of the AFD RGCs are capable of conferring thermosensitivity to other C. elegans neurons in a tax-4 dependent manner. So here I'm plotting calcium responses produced by the RGCs that have been misexpressed in a C. elegans chemosensory neuron called ASE. And so what I want you to notice is that between these 2 genes, each one confers sensitivity to a different temperature range. What about the stercoralis homologs? So for these plots, I'm again showing average calcium response of C. elegans ASE neurons misexpressing each of the stercoralis AFD RGCs. So 2 things. First, all 3 stercoralis RGCs are conferring thermosensitivity compared to only 2 in C. elegans. And the second thing to notice is that whereas in C. elegans, only 1 RGC, GCY18, confers sensitivity to worm temperatures, all 3 of the stercoralis AFD specific RGCs are responding to body heat. So to conclude, my research demonstrates that IL-3s display 2 parasite-specific modes of temperature-driven behavior, including attraction to human body heat. I found that parasitic and free-living nematodes rely on the same sensory transduction pathway and inputs from the same sensory neuron pair to drive their divergent behaviors. And I presented calcium imaging results that suggest that stercoralis thermosensory neurons more strongly encode temperature ranges that span human body temperatures. And I identified one possible mechanism, an expanded number of thermosensory proteins in Strongyloides stercoralis that confer sensitivity to human body heat. And this contrasts with the situation in C. elegans and suggests that changes in primary sensory neuron responses may be contributing to the ability of parasitic nematodes to seek out hosts using heat. And the last thing I want to mention is that taken all together, these results are actually the first direct evidence of differential sensory encoding by homologous neurons in parasitic and free-living nematodes. And with that, I'd like to thank all the members of the Hallam Lab, but particularly the 3, at the time, pre-graduate researchers who've worked with me on this project over the years, Feli Ruiz, Junha Lee, and Jackie Lopez, as opposed to the funding that pays me, and all the Neuromatch folks.
Thank you very much for your talk. It was really nice. We have very small time for some questions. So actually, my question has to do with, as far as I'm really naive to this topic. But as far as I can see, there is a preference towards the temperature, but then there's also the motion, like the speed of the motion towards, like, for example, when you were showing this graph with the AFD silencing, I could, at least from what I could see, there was also a very big decrease in the motion or in the speed. Like how far, like the.
Yeah, so there's 2 things. I guess that it's known. So we observed that as parasites, the IL-3s engage in host seeking, like are doing positive thermotaxis, the speed of their movement actually increases, and that seems to be due to the sensory drive provided by the temperature cues drives them to move quicker. There's also, you might have noticed a difference in the directionality, I guess, of their movement when they're not engaging in heat seeking or not engaging in temperature driven migration. They sort of move around in little curls, like they're very circuitous. They straighten out in a linear thermal gradient in a way that's extremely dramatic, and we don't see when they respond to other sensory modalities. We don't know why they do that.
Interesting. Okay, so I don't see any other question, and we are kind of past only 1 minute from our time. We're still fine. I would really like to thank you for your time and for this amazing talks. And I hope to see you in the next Neuromatch meeting. Thank you very much. Bye-bye.